My presentation will be on extractables testing of bioprocessing materials. I will give some practical examples and also some of the hurdles you might encounter when performing extraction studies on process materials. So first, an overview on the design parameters you should take into account when doing or designing extractable studies for the process materials. Then a first case study, which I call a model study. So all of the design parameters are in line with the standard testing protocols. And here we will go to look at autoclaved versus gamma irradiated filters that were tested. In a second case study, I'm going to talk about an incompatible study setup and how we dealt with that. A third case study is on downscaling of process items to make them compatible with laboratory testing. So a downscale of the large test item to a smaller test item. And case study 4 and 5 will be on the monitoring of solvent loss on bag systems and on filters. So first, the design parameters to take into account on extraction study of process materials. I've listed the main parameters in the first column and then the values that are stipulated in USP 665 and Bioforum standard protocols in second and third column. With regard to extraction solution, mainly of importance for organic content and pH, we can see the 50% ethanol water extraction solvent and aqueous solution at low and at high pH for both testing protocols. For Bioforum, there is an additional solvent being water without pH adjustment, and for some drug products of processes that can have some added value if your product is at neutral pH. For both testing protocols, the extraction temperature is 40 degrees Celsius. With regard to extraction time, USP 665 has 3 time points, 24 hours, 7 days, or 21 days, and only one time point should be tested according to that protocol. With regard to BPOG, there is an additional time point, 70 days, so that could be interesting if you have long storage applications to take that also into account. And also this guidance recommends for most of the process components at least to also test multiple time points. With regard to surface area to volume ratio, 6 square centimeter per ml is applicable for both testing protocols for all materials except filters, where 1 square centimeter per ml is recommended or higher. And that's mainly talking about the effective filtration area of the filter material inside the filter housing or the filter cartridge. Extraction methods are rather a filling or a submersion of the test item, and both testing protocols also recommend to do a dynamic extraction. This can be done by a shaking, incubation, or by a recirculation. So the first case study I want to discuss is the model study for a moderate risk component based on USP 665. We are going to go through that example by comparing results of an autoclaved and a gamma irradiated filter. And as you can see, all the design parameters are in green, meaning that we are all fully aligned with the recommendations of the standard testing protocols. So first, when we're designing an extraction study, we would require sponsor information in order to have all information available to come up with a good study design. The first information is on the process material itself. So here we're looking at a polyether sulfone filter. Of at least the membrane is made of polyether sulfone. It also has a polyester support, and it's encapsulated in a polypropylene housing. Another important information is the effective filtration area. In this example, it's 6,000 square centimeter, and that will allow us to report results in microgram per square centimeter. If the sponsor has, for instance, filters made of the same materials of construction, but in a different size, the results can also be extrapolated to the appropriate filter size in the process. Another important parameter are the sterilization conditions that should be taken into account when doing the testing, so that is the same as in the actual process. In this example, the sponsor wanted to test both ways of sterilization that are commonly used. So autoclaving and gamma irradiated were both tested in the study, and also results will be compared. Besides information on the process component itself, we also require the conditions as it is used in the process to have a good understanding how it is used. In this case, it is at 30 degrees Celsius with a contact time less than 24 hours. The minimum homogeneous process volume in this particular process was 20 liters, and the filter was used to filter an aqueous solution with a small amount of drug substance, about 10% of sucrose. It was in an organic buffer with almost neutral pH, and a small amount of surfactants were also present. So taking into account that information, we designed following study, and it was mainly based on USP 665 because it was a moderate risk component. Extraction was performed using filling and shaking incubation, and we selected the short-term contact conditions of both testing protocols, being 40 degrees Celsius, 24 hours. And when we compare them to the actual process conditions, you can see that they are definitely worst case. 50% ethanol in water is recommended for medium risk testing. Also comparing to the actual process solution that is filtered, this is also definitely worst case. The extraction ratio, in this example, we have filled the filter entirely and then closed it at its open end using inert materials. So we are really dependent on what is the fill volume of the filter. In this example, it was 1.5 liters, taking into account the effective filtration area. We had an extraction ratio of 4 square centimeter per ml, which is higher than the 1 square centimeter per ml recommended by the standard testing protocols, and also definitely worst case compared to the actual process. Once we have generated the extract, we have to make it compatible with our analytical methods. In this example, we had to perform liquid-liquid extraction to dichloromethane for analysis by GCMS and LCMS screening. And for this study, we used the organic screening methods for volatile, semi-volatile, and non-volatile organic compounds because we wanted to find out which compounds are potential leaching from those filters. In order to evaluate at a wet level, we should look into the chromatograms. We calculated an analytical evaluation threshold. As already explained by Karen, you first start with the toxicological threshold. In this case, it was 1.5 micrograms per day because it was for a parenteral application. We take into account the maximum daily dose of the product as administered to the patient. In this example, 1 ml per day. And then we also should take into account the process information. So the minimum volume that was filtered in the process is 20 liters, and the filter has an effective filtration area of 6,000 square centimeter per filter. Doing the math, we end up with an estimated IAT of 5 micrograms per square centimeter, and then taking into account our uncertainty factors, we have final AAT levels of 2.5 micrograms per square centimeter and 1 microgram per square centimeter for LCMS. And for the study, then we have to make sure that the reporting limits are set below those final AAT levels, just to make sure that we report all compounds that should be subjected to a safety evaluation. So first have a look at the Headspace GCMS screening results. The screening methods are mainly intended for, or the focus is on the discovery and the identification of the compounds that are migrating from the test items. And these are differential analysis, meaning that we compare the extract of the filter in this example compared to a blank solution, which is actually the extraction solvent that has not been in contact with the filter. And only compounds that are above the reporting limit will then be reported. So in the chromatograms, you can see at the upper tray the chromatograms of the filter extract for autoclaved and gamma irradiated filter. At the bottom is the extraction blank, and all differential peaks above the reporting limit will be reported. They are summarized in this table. I will go into more details on the results later on during this presentation. At this moment, I just wanted to indicate that we have more components for gamma irradiated filter compared to the autoclaved filter, as you can see in the chromatograms as well in the summary table. For GCMS, we did the same exercise, and comparing the results, we can have the same observation that there are more compounds detected for the gamma irradiated filter, but also that additional compounds were detected compared to the Headspace GCMS analysis. And this is already an illustration that it's important to use complementary analytical screening methods to cover the whole spectrum of potential extractables that can leach from process materials. For the non-volatile organic compounds, LCMS was used using APCI in positive mode, where we have unique compounds for the autoclaved and the gamma irradiated filter. And again, additional compounds were detected compared to the screening methods for GC and headspace GCMS. And also LCMS in the APCI negative was performed, giving again additional compounds to the positive mode. So also within the technique, complementary ionization modes should be performed. At this table, you have an overview of all compounds that were detected for the autoclaved and for the gamma irradiated filter. And when we compare the results compared to the final AET, no compounds were detected above final AET by headspace GCMS. Also, no compounds were detected above final AET by GCMS. And for LCMS, there was one compound above final AET, and this compound should then subjected to a toxicological assessment, or a subsequent leachable study could be performed in order to evaluate if this compound is also detected using the actual process solution and actual process conditions or not. The results also have some information on understanding what is the difference between an autoclaved and a gamma irradiated filter. First, we will look at the compounds that were detected for both cases. There were some compounds that could be related to the filter housing made of polypropylene. There were also some compounds related to the filter support made of polyester. Some compounds could be related to the filter membrane made of polyether sulfone. And some compounds are related to antioxidants or degradation products from those antioxidants. And there were also some processing aids detected in this, or for both cases of the gamma irradiated and the autoclaved filter. Looking at the unique compounds, we can also have some interesting observations. For a first compound that was only detected for the gamma irradiated filter was a cyclic siloxane. And we could trace back that this was because the filter that was subjected to gamma irradiation was part of an assembly that contained silicon tubing. So at the moment of irradiation, potentially some of the silicon tubing components migrated to the filter. And as a result, when we do the extraction only on the filter, it can still appear as an extractable from the filter. The autoclave filter was autoclaved as such, so it's not part of an assembly, and there this compound was not detected. Another set of compounds that were detected were related to solvents, and they were only detected for gamma irradiated filter. So potentially during the autoclaving of the filter, the solvents were removed because autoclaving is at a rather high temperature of 121 degrees Celsius, which could result in evaporation of those solvents from the filter during that step. And a third set of compounds are related to antioxidants. And as you can see, for the autoclaved filter, only Erganox PS800 was detected, while for the gamma irradiated filter, some other compounds were detected. If you look into more detail at the bottom right of the slide, you can see the structure of Erganox PS800. And when you gamma irradiate that component, it can degrade into some degradation products, which were exactly the compounds we have detected for the gamma irradiated filter. So there you can see that the antioxidants are doing their job in protecting the polymer by degrading into their degradants and therefore not degrading the polymer. And also the same for the 1,3-D-tertiary-butylbenzene, which was only detected for the gamma irradiated filter. Also, this is most likely formed by gamma irradiation from the antioxidant Ergafos 168, which is commonly used in polypropylene materials. So that was the first case study, the model case study. Now we are going to look in some case studies where we did some deviations compared to the standard testing protocol for whatever reason, let's say. And the first I want to discuss here is an incompatible study setup that we have encountered when testing a transfer hose. So the transfer hose was made of a polyurethane material and stainless steel. It was a very large test item, 6-meter hose that we have tested, so resulting in a very large contact surface area. Sterilization conditions were not specified by the sponsor, they just indicated that they use it as supplied by their vendor. Process conditions for this hose during the use and the process are maximum 30 degrees Celsius with a contact time of less than half an hour. And the hose is used to transfer a drug substance powder into a large processing vessel, and the powder is actually a solid material, but after transfer of the powder there is also transfer of medium chain triglycerides, which are actually a liquid, and because of that we took this test item also in consideration for testing according to USP 665. The minimum mass after the transfer of the ingredients to the large processing vessel was 2,500 kilograms, and the product that's made is actually a drug substance in medium chain triglycerides, so a topical cream which is composed of 100% of organic ingredients. For the study design, we again used the filling and shaking incubation method, where the tubing was entirely filled with the extraction solvent. We used in the first trial 50 or 40 degrees Celsius 24 hours, so the standard conditions for short contact materials. They are definitely worst case to the process conditions, but we decided to go with them because they are stipulated in USP 665, and you cannot do less worst case testing as stipulated in the document, at least at first. With regard to the extraction solvent, we selected, or we decided to go with ethyl acetate, and that selection was done based on the dielectric constant, so the process solution in contact during the actual process were the medium chain triglycerides with a dielectric constant of 3.9. Looking at a compatible solvent with our analytical methods and having a dielectric constant close to that value, we ended up by using ethyl acetate as the extraction solvent. Going with 50% ethanol and water, as you can see, was not a very good choice in terms of a mimicking solvent, because those electric constants of water and ethanol are quite far away from that of the medium chain triglycerides. The extraction ratio in this example is 1.1 square centimeter per ml, so not the 6 square centimeter per ml. That's because we decided to fill the tubing entirely. The sponsor could not provide us with the tubing with a smaller inner diameter, which can be used to increase the extraction ratio, but that was not an option for this example. And also we wanted not to cut the tubing into pieces, because that would also not be a representative contact compared to the tubing as it is used in the process. With regards to sample preparation, the ethyl acetate can be directly injected into our instruments. And again, we use the standard screening methods for the volatile, semi-volatile, and non-volatile compounds. So when we started the extraction, everything looked fine, as you can see at the picture at the left. Then we applied the conditions, checked before the end of the evening if everything was fine, and it was still okay. But in the next morning, we ended up with this, which does not look as a successful experiment. There were some incompatibilities between the solvent, the temperature and time, and the test item. So it's definitely clear that we need to revise the testing protocol, even though extractable could be recovered for testing. So taking into account some additional information, we decided to go still with the filling and shaking incubation method, but to change the standard testing protocol contact conditions of 40 degrees Celsius 24 hours to 30 degrees Celsius for only 1 hour. So we are still worst case compared to the process, because at maximum 30 degrees Celsius, maximum 30 minutes. So that should still be okay in that condition. Ethyl acetate was still selected as the extraction solution, because we were convinced that's the best mimicking solvent to use, and therefore also providing the most relevant extractables data. And again, filling and shaking incubation was used. So at the beginning, the same experiment was performed, now in a glass vessel, to be sure that there are no any leaks during this process. After these conditions, luckily we found an intact test item afterwards, and we could recover extract successfully that could be analyzed on the analytical instruments. Also for this project, an analytical evaluation threshold was calculated in the same way, using the safety concern threshold, the maximum daily dose, the information on the process the maximum weight of minimum weight that is collected after the materials have passed through the hose and the surface area of the hose. We ended up at an estimated AET of 34 micrograms per centimeter, and we took into account our uncertainty factors for the final AET levels. Reporting limits were set below those final AET levels, and we ended up with these results. So for headspace GCMS, no volatile compounds were detected above the reporting limit. For GCMS, you can see we ended up with 12 compounds to be reported. The ones highlighted in red were above the final AET level, so their toxicological safety assessment should be performed. And also for LCMS in positive and negative mode combined, we ended up with 25 components, of which only the one in red should be subjected to a toxicological assessment. A third case study I wanted to present was a study we have done during the COVID period, where it was difficult for some of the suppliers to provide the pharmaceutical companies with process items at a laboratory scale. So they were mainly focusing on producing test items at a process scale, let's say. And the sponsor wanted to continue with the testing anyway. So in agreement with them, we made a small scale version of their process scale test item. So in this example, we tested a 100-liter bag system that is used for the storage of a buffer solution. The bag is made of an LDPE film, and it also contains platinum-cured silicon tubing as well as polycarbonate connectors. Because it's a very large bag, of course, it also has a very large surface area and definitely not compatible with our laboratory incubators. So we had to make a small-scale version of that. Sterilization was by gamma irradiation at typical conditions that we see for process materials. With regard to process information, the bag is used at maximum 8 degrees Celsius and has a maximum contact time with the product for 10 days. The buffer solution at a neutral pH and an organic content of about 12%, mainly because the presence of sucrose, and the bag's minimum fill volume during the process was 30 liters. So taking into account that information, the extraction study was designed again using filling and shaking incubation, but on a small scale model. A few words on that in the next slide. With regards to extraction temperature and time, we went with the 40 degrees Celsius as extraction temperature, but the extraction time was calculated based on the ASTM guidance for accelerated aging, or also the Arrhenius equation. And by using 8 degrees Celsius 10 days, we found out that using 48 hours would be a worst case compared to that process conditions when we do the testing at 40 degrees Celsius. Extraction solvent was 50% ethanol and water again. This was also a worst case compared to the process solution, up to 12% of organic content, neutral pH. And the extraction ratio was close to 6. We went with a little bit lower because we wanted to make sure that the extraction solution was in full contact with all contact materials of the bag system that we had made from the process scale bag system. Doing the sample preparation, and then we could do the analytical screening methods for volatile, semi-volatile, and non-volatile organic compounds. In this slide, I want to talk a little bit more on how we handle to downscale the 100-liter bag system into a laboratory scale bag system, let's say. In order to do that, we first identified the different components of the 100-liter bag system. So we have the bag film, the tubing, flanges, connectors, teeth, and clamps. Once we have that list available, we calculated the contact surface area of the main components, so for the bag film and the tubing. And then for making the small-scale bag system, we, by trial and error, find a scaling factor, let's say, that we could use to make a bag that is still compatible with our laboratory incubators. And for this project, the downscale factor was 6, meaning that the surface area of the 100-liter bag system was divided by 6 to have the small-scale bag system, and we did the same scaling factor for the tubing. So we made the tubing shorter to give it the same contact surface area as in the actual process. For the other components, which are only in contact with the process solution in very small amounts in regards of surface area, there was not an option to cut a connector in 3 or in 6, let's say. So there we decided just to go and to test them as such, and understanding that if we extrapolate results in micrograms square centimeter for the small scale bag system to the large scale bag system, results or extractables coming from those components would be overestimated because they were not scaled down. So using that scaling information, the lab team managed to transform a 100-liter bag system to a 450 ml bag system that could easily fit our incubator. So they literally cut out a piece of the bag film and sealed it at the open ends using a heat sealer. Also, the tubing was cut and shortened to have the same surface area, which is 6 times smaller than in the actual process. Also, connectors were included and connected to another, so as they are used in the process, and the clamp was used at the end of the tubing to make sure that we have a closed system to do the testing. Also, for this study, an analytical evaluation threshold was calculated doing the same calculations as we have described before. Here we are ending up with a final AET of 2.5 micrograms per square centimeter for headspace GCMS and GCMS, and 1 microgram per square centimeter for the LCMS analysis. And for this project, no compounds were detected above a reporting limit of 1 microgram per square centimeter, and the sponsor was very happy that we managed to scale down their bag system and to assist them in the challenging times with the result that they could safely use that process bag in their production. A fourth case study is on monitoring of the solvent loss, and I want to discuss this first for the bag systems that are typically used for storage applications and therefore in quite long contact with extraction solvents and process solutions. This example here is actually for a cell culture bag that was requested by a supplier of the bag. They managed to provide us with a small scale version of the bag, so we have no issues on the scale of the test items, but they were not very open, let's say, on the materials of construction or sterilization conditions that were used. So they were very protective on that. The only thing they informed us of was the surface area of the bag system. So when we report results in microgram per square centimeter, they could easily recalculate results to the process dimensions that is used in an actual process by their customers. They requested to use the bioforum testing protocol, and they wanted to cover long-term storage as well. And they had also a specific request to make sure that there was one test with minimum contact between the extraction solvents and the tubing, and one with maximum contact between the extraction solvent and the solution. So in practice, that's only setting the white clamp on the picture at a different position. So minimum contact, the clamp is close to the bag film, and with maximum contact, the clamp is at the end of the tubing. So for the extraction conditions, filling and shaking incubation was used. The 3 or the 4 BPOG solvents were selected, and the extraction time was 70 days at 40 degrees Celsius. Extraction ratio 6 square centimeter per ml. Because we did not know the materials of construction of the bag system, we were a little bit suspicious, let's say, with regards to solvent loss due to evaporation, because we commonly encounter, especially for 50% ethanol extraction solvent, that there is a considerable solvent loss for some of plastic materials. So in order to deal with this, we decided to measure the weight of the filled test item at different stages during the incubation. And at this slide, you can see the results for the 50% ethanol extraction solvent and the aqueous extraction solvent. And the first observation is that you can see that the solvent loss is almost a linear equation. So the longer you store a test item, the more risk for solvent loss. It is quite straightforward. And also the 50% ethanol solvent gives a higher solvent loss than the aqueous solution. So that's still in line with expectations. And therefore, we also managed to do an extraction with a solvent loss below 20%, which is specified in USP 665. And that information is also needed to calculate the results that we measure on our instruments in micrograms per liter to micrograms per square centimeter, taking into account the fill volume of the bag at the beginning of the experiment. The situation was a little bit different when we're looking at the results for maximum contact with the tubing. There you can see that after already 20 days, there was a risk for the 50% ethanol to be too much of a solvent loss during that incubation. If we made the extrapolations from the results we had so far, it was clear that when we reached the incubation time of 70 days, the solvent loss would be higher than 50%, and then USP 665 stipulates that we should repeat or modify the study's design just to make sure that the solvent loss is less than 50%. So in agreement with the sponsor, we decided to remove the extraction solution from the tubing after 20 days of storage and proceed with the incubation to 70 days with all extract solution inside the bag. So without contact with the tubing, and by doing so, we ended up with a solvent loss less than 20% for the aqueous solution, and for the 50% ethanol, we managed to have a solvent loss between 20 and 50%. And in that case, USP 665 recommends to use the recovered volume, so the volume after the extraction, to do the calculation afterwards to micrograms per square centimeter. In the final case study, some words on the solvent loss by filters, because different things are going on when we're doing extractions on filters compared to bag systems. To illustrate this, we have taken the example of a filling and shaking incubation of a filter that was stored at 2 time points, one at 40 degrees Celsius 24 hours, and a second time point after 40 degrees Celsius at 21 days. In this study, the USP 665 solvents was used, and we again used the entire filling of the filter, ending up with an extraction ratio of 5 square centimeters per ml. Looking at the results that we have obtained by monitoring the solvent loss by volume, you can see that for the aqueous solutions for the 24 hours as well for the 21 days time point, the solvent loss was about 20%, and for the 50% ethanol solution, you can see it was about 28%. If we then look at the USP recommendations, we're in the situation 2 where we should use the recovered volume, so after the incubation, to do the calculations, but we were a little bit suspicious because the time point between 24 hours and 21 days have almost exactly the same numbers, while you would expect that solvent loss by evaporation at least would increase over time, as we have seen for the bag system. So is the assumption correct that the solvent loss is because of evaporation, and should therefore we use the recovered volume instead of the initial volume to do the calculations? In order to find out, we also monitored the solvent loss by weighting the filter during different stages of the extraction process. So the first measurement we did of the empty filter, so without contact with solvent. Then we measured the mass of the filter and the solvent at the beginning of the incubation. We did the same after the end of the incubation, and then of the empty test item of empty filter after we have recovered as much of the extract as possible. Looking at the results by weight, we can see that we are in the same line as we have found based on the solvent loss by volume. But by using the weights, we could also break down the solvent loss in a solvent loss by evaporation and a solvent loss by absorption. And then it was very clear that most of the solvent loss that we have seen was due to absorption of the extract on the filter membrane itself, and almost no solvent loss could be assigned to evaporation of the solvent during the incubation. So there are different mechanisms going on, and because the extract is not concentrated due to evaporation of the solvent loss, I think it's better to use the initial fill volume to calculate the results in case of significant amount of absorption, just to make sure that you're not underestimating the concentration of the extractables due to an incorrect assumption of solvent loss by evaporation. Thereby we have come to the end of the presentation.